Hey y'all, my name is Prashant Kumar, or PK, and I'm an MCAT tutor at Shemasian Academic Consulting. Today, we're going to be doing a passage walkthrough in the MCAT Biochem section. You should be taking roughly 6.5 minutes on this passage, but today, we're going to go more in depth so you can see exactly what's going on in my head as I tackle this section. Let's get it. Alright, great. So I've gone ahead and put the passage on the left and the questions we're going through on the right. So feel free to take a moment and pause the video and come back to us when you're ready. If not, let's get right into talking strategy. For me, I like to maintain the same exact strategy regardless of whatever passage I go through. The reason I like to do this is that it gives me something to ground myself, regardless of whether the passage is super easy or super challenging. I know I'll have something to ground me. This boosts confidence, and oftentimes confidence is the most important thing on test day to stay comfortable with the test and keep moving at a reasonable pace. For me, my strategy is read a quick paragraph, write a 3 to 5 word summary, go on to the next paragraph, and on and on and on. The reason I like to do this is that it provides accountability. At the end of that paragraph, I need to know what's happening in it before I go on to the next paragraph. It prevents me having to reread an entire passage, and instead, if I don't understand a paragraph, I waste a little less time just skimming through that paragraph again, making sure I know it before I go on to the next one. The 3 to 5 word summary is a great accountability check, and I highly recommend trying it if you haven't already. With that, let's get right into the passage. Oculocutaneous albinism, OCA, is a genetically heterogeneous group of disorders characterized by absent or reduced pigmentation of the skin, hair, and eyes from the time of birth. OCA type 2, OCA2, is the most common form of the disease, but no effective treatments currently exist for the disease. Researchers interested in studying OCA2 decided to develop a mouse model of the disease as a model through which potential therapeutics could be evaluated. Alright, so the question I always ask myself at the end of reading this paragraph is, what's happening? In very brief terms, what's happening? And so if you write that down in 3 to 5 words, all I'm going to say here is OCA disease results in X treat, right? And then I'm going to point to mice. Something quick. It doesn't have to make any sense to anyone except you. As long as you're able to read it, no one else is going to be reading your notes, right? And so having this brief sort of abbreviation, put arrows, put symbols, whatever you need. And with that, we're ready to go on to the next paragraph. To identify the causative gene in the Z0015 mouse cell line, researchers performed a family-based genome-wide linkage study, GWLS, to map the chromosome regions co-segregating with the mutant phenotypes. The results showed a single missense mutation, C2228, C is greater than T, that resulted in a serine to transition to leucine. OCA2 S743L, S743L, hereafter referred to as S743L, in exon 21. Further data revealed that the S743L mutation was likely the causative mutation for the OCA2 in mice. This mutation is orthologous to the human S736L mutation. Alright, so the question happens again. What's happening? And right here, we have a lot of details, right? We have a lot of information, some abbreviations, some locations, things like that. And I always say, don't focus on the details on this first read. Focus on the big picture and use it like a table of contents. If we need to come back to it, we know exactly where to come back to. But here, we're going to type, what are we going to write? We're going to say that this S743L mutation is like important to OCA2, okay? That's all we're thinking, right? This is a big picture understanding, and that we don't need to focus on all these specific details, whether they're co-segregating, I don't even remember what orthologous means, right? Things like that you're not gonna have remembered from your content phase, nor that you're gonna have remembered simply from reading the paragraph once. And so we'll come back to it if needed, but right now let's go on to the next paragraph. The S743L mutation is located in the predicted transmembrane domain 10 of the porcine OCA2 protein and is evolutionarily conserved among distinct mammals, indicating its vital role within this domain for the function of OCA2. the S743L mutation did not alter OCA2 expression at the transcript level. Researchers then hypothesized that protein expression might be affected, and they measured protein expression in the eyes and scalp of mutant pigs. The results are shown in Figure 1. All right, so right away we see a figure. And another high yield tip I think I have is don't spend too much time on the figures on that first read. The reason for this is that you don't know if a question is going to be asked or what information is going to be asked about from a figure, right? On that first read, all I want you to do is be comfortable with it. Like look at it, see if you understand basic idea of what's going on, and that's it. And then we'll come back to it if needed. But with this, let's just do our quick 3 to 5 word summary first. And so here it's saying S743L, it does not, right? I'm going to draw a little X, protein or transcript, right? It does not alter transcript. So I'm drawing an arrow with an X through it. And so I'm going to write protein level, and I'm going to put a question mark right there. So this is my way of saying, like, we're looking at whether it's modifying the transcripts or modifying the protein level. And so having this sort of distinction is very important because it tells us straight away that there is no effect on the transcript level. All right, and so let's look at the figure real quick. And oftentimes I just take a look at what they're saying in this little caption. And so it's looking at the OCA2 expression level in S743L mutant mice by Western blotting. And so when we look at Western blotting, right, we know that's a type of blotting that looks at proteins, right? Snowdrop, if you don't remember that, take a look at some of the Shamassian articles on how to remember these different blotting techniques. But here we are having a Western blot of some sort with different sort of locations in the eye and the scalp of mutant pigs, right? I'm not going to spend too much time looking at the figure, but we're just going to go on to the next paragraph. Next, researchers sought to investigate whether S743L mutation induced growth or fertility aberrations in OCA2S743L, S743L pigs. Researchers characterized the growth and reproductive traits of the pigs in comparison to wild-type pigs by measuring litter size, the number of weaned pigs, and mean litter birth weight. The results from this set of experiments are shown in Figure 2. All right, so again, what is happening? Big picture understanding. S743L versus wild type on litter, number, and weight. Okay, so that's like exactly what we see right here in this figure. I don't want you to spend too much time looking at it. Total litter size, number weaned, and birth weight, right? And it says that exactly the same thing in Figure 2. And so with that, we've done it. We have analyzed this entire sort of passage to understand what is the big picture idea, right? We have genes, we have sort of experimental designs, graphs, all of that. And so with that, we're ready to go right into question number 1. All right. The change in properties of amino acids in the causative mutation for OCA2 can be best described as changing A, charged amino acid to a hydrophobic amino acid, B, polar uncharged amino acid to a hydrophobic amino acid, C, hydrophobic amino acid to a polar uncharged amino acid, or D, aromatic amino acid to a charged amino acid. All right, so with that, we have the question, we have our answer choices, and we need to remember exactly where we're looking, right? We're looking at where it first talks about this sort of mutation and what's happening in this mutation, right? And so we know that the mutation is written in this form, right? S743L. And this is a common format to talk about amino acid changes, right? This is going to be your original, and this is going to be your final, what it's changed to, and this is going to be your location, okay, of whatever mutation is happening. So right away, I need you to know your amino acid abbreviations by heart, the 1-letter and the 3-letter abbreviations, and exactly what they look like. This is high yield for the MCAT. But even if you don't know it this time, you're lucky. They have given you exactly what is happening, right? It's telling you that you have a serine transition to a leucine right there in that second paragraph. So with that, we have a serine being modified to a leucine. And so right away, I need you to know what the side chains of these look like. And so a serine will look like OH, right? And so what does a leucine look like? A leucine looks like right? So that's leucine. And so with that, we should realize this is a very obvious change of one type of amino acid to another. We should know serine is our example of a polar amino acid. And we should know that leucine, since it's a bunch of strings of carbons, is our example of a nonpolar or hydrophobic amino acid. Okay? And so with that, we can already say this is a polar amino acid and this is not charged. Neither are charged, right? Both of these are not charged. And so we can eliminate A because we're not dealing with a charged amino acid. C, because our original is serine, which is not a hydrophobic one, is a hydrophilic one. Or D, aromatic. It's not aromatic. So already we're able to pick B, polar uncharged amino acid, to a hydrophobic amino acid. And we crushed it. We're 1 for 1 and on to the next question. So I really can't emphasize enough, gotta know your amino acids. They're gonna come up all over the test. All right. Question number 2. Based on figure 1, which of the following conclusions is most likely to be correct? Is A, the S743L mutation decreases OCA2 transcription. B, wild-type mice contain higher levels of OCA2 transcripts. C, S743L mutants have a lower levels of OCA2 protein. Or D, wild-type mice translationally upregulates OCA2 protein. All right, so when we're looking at protein and transcripts, right, the words that come up over and over again, and it tells us exactly where to look as well, figure 1, we know where we're going to look, right? We're coming back to this figure and this paragraph. And so right away, we want to look at these blotting techniques as a comparative measure, right? So we have 2 different sort of sets of data. I'm going to draw a little line here. We have the eye, which is set 1, and we have the scalp, set 2, okay? And beta-actin is a commonly used control to measure how much of the substance we're putting in there. And so it's like a standardizing control to make sure our experimental setup is perfect. Just want to make sure you understand that beta-actin is not really what we're measuring in our experimental design. Rather, it's a control to make sure our experiment is set up perfectly. All right, so now we are looking at OCA2, right? That was what the experiment was supposed to look at. And we're looking at this wild type and S43L. And so I want you to use this as in the same way in both of these experimental designs, right? We have in the scalp and the eye. So what do we see from the wild type to this mutant, right? We have this dark band and it's getting lighter, right? Big picture, that's all that's happening. It's a dark band, very thick, getting lighter. Here on the set 2, on the scalp, it's the same exact thing, right? We see that we have this darker band, like lightly darker, turning into this thin, like nothing band, right? You can barely see it. And so we're seeing a lower sort of expression of something, right? And when we said Western blot, what are we dealing with, right? There's the mnemonic snowdrop, and snow Western equals protein, right? Highly recommend reading up on snowdrop if you don't remember it, but Western blot means protein levels. So already we know we're focusing on protein levels, and we know that S743L is decreasing the amount of expression of this protein level, okay? And so with this, we can already say, oh, we have protein, right? We know 2 answers are dealing with protein generally. So how do we know it's not dealing with transcription, right? How do we know it's not dealing with that? Well, we said that right in our little summary, right? It tells us in the paragraph that it doesn't affect transcription, right? OCA2 expression at the transcript level was not altered, okay? And so right away, we can eliminate A and B, and we're looking at C and D. So the determination is whether S743L mutants have lower levels of OCA2 protein. That sounds pretty good, right? We saw a lighter band in those mutant variants, indicating that there was less protein available to be seen. So C is looking pretty good right now. D, wild type mice translationally upregulate OCA2 protein. It doesn't say anything here about translationally upregulating protein, right? The wild type is just maintaining its same regulation of protein. However, the S743 has some sort of mutation that affects the protein expression. So we can eliminate D, and we're left with C, right? And so with that, we're on to question number 3. All right. Researchers find a second mutant that produces a UAG codon in the DNA. Which of the following best characterizes the nature of this mutation? Is it A, nonsense, B, point, C, frameshift, or D, missense? All right, so this is a standalone question, one we don't have to go back to our passage through, right? It tells us that we're looking at this UAG codon in the DNA, and what is the type of mutation that comes from a UAG codon? And this is a fact we need to know. A UAG codon is a stop codon, right? It's a very, very important stop codon. And I highly recommend you remember this term, stop codon. It can also be called an amber codon, right? Something that stops it prematurely. And so what does a stop codon do? It ends all of the sort of transcription translation process because you don't have anything that you can translate, right? You have no sort of no codons that can match it other than a stop codon. And it ends translation. And so with that, what do we call something that translates into a stop codon too early, right? So it's not a missense, right? A missense is a change in amino acid because of one sort of mutation, right? So you could have, instead of sort of a serine, you might have a threonine, right? Something like that. You just have a shift of one amino acid to the other one. A frameshift mutation is oftentimes very dangerous as well. A frameshift mutation is an example of sort of you have A, and all of a sudden you have a G inserted there, right? You have a G inserted there, or another letter, and that changes the frame shift. So the reading frame turns from these three to these three, and then puts it off by one in this case. So we're not having a frame shift mutation. A point mutation is a type of mutation, right? It's a type of changing one letter. But in this case, our nonsense mutation is our correct answer. Since we're implementing a stop codon too early, right, this UAG codon, that means we're dealing with a nonsense mutation. So we're changing our amino acid to a stop codon sequence, right? And so it doesn't tell us whether the second mutant is a point mutation or not. It could be, it could not be, so we can eliminate B since it's not given to us. And so since we know it's a stop codon too early, we're dealing with a nonsense mutation. All right, onward to question number 4, our final question of today. All righty. Oops. Okay. A researcher concludes that S743 mice produce heavier offspring than wild-type mice. Is this conclusion reasonable? A, yes, S473 mice produce a litter with a higher birth weight. B, yes, S473 mice without functional OCA2 have lighter organs. C, no, the difference in mean offspring weight between wild-type and S473 mice is not significant. D, the data in figure 2 contains too many outliers to make a conclusion. All right, so again, we know that we're looking at a certain figure. We have to determine what figure we're looking at, right? When we're talking about sort of these S473 and heavier offspring, which figure are we going to look to, right? Figure 2. We should remember that one of the things we looked at in figure 2 was our weight. And so we're looking at this one, this diagram right here, dealing with litter weight. And so first and foremost, I want to talk you through how to read this graph, right? We have this wild type, we have this S743, and then we have sort of a plot with our mean litter weight on the y-axis. Hone in on those axes and use it to tell us information that we don't know. So here we have a bunch of different mice as these black points, determining our sort of weight, our the weight of the litter. So here we have mean litter birth weight at different values on the wild type side and the S743 side. And so for there to be a significant difference, right, before we even go into the answer choices, for there to be a significant difference, you would have to be able to tell something, the values, to look separated beyond sort of their standard deviation. So in this case, this is the sort of mean value or the center value, and these sort of windows that they draw here based on this line and this line are the standard deviations, telling you how far wide the width of these sort of the range of these values are. And so with that, we want our windows to be completely separate from the wild type and the S743L window for you to be able to sense that there is a change. So first and foremost, to determine whether there's a change, the windows need to be completely separated. They can't have any overlap. And so that's very, very important. And so we can see here that even if we draw a line through the windows, there's definitely overlap here. So first and foremost, no, there is no significant change. That's very, very important. Another indicator is that we have not seen any stars. Another way they draw it is if they have stars above here, right? And we do not have any stars drawn in here. And so the stars would indicate whether there's significance with a p-value less than 0.05. Make sure to check out your statistical knowledge tools again. But here we know that they look very similar, right? Just because the mean is off a little bit, it could be due to random chance or random error. And so we note that there is nothing significant happening. We can't tell if there's an actual experimental difference. And so right away, we can eliminate A and B, right? We don't know if they're producing heavier offspring or not because they're within that window of chance. C, know the difference in mean offspring weight between wild type and S473 mice is not significant, is looking like a great answer choice because of that, right? We don't know. It could be, it could not be. Some cases could be, some cases cannot be, right? And that means that we don't know. And that's fine. That's going to be a lot of experimental setups. We're not going to know exactly what's going on with everything. And then finally, D, the data in figure 2 contains too many outliers, right? It doesn't really, we don't know anything about outliers, right? There's a couple of outliers here, but we have a window to work with. And with that window to work with, that's all we really need, right? We can tell significance based on that window, or if they give us the stars up top. And so we can eliminate D. The outliers don't affect that. And we're 4 for 4. We crushed it. All right, great work, team. We crushed another biochem passage. Kudos for you for getting it done. Lots of great material, from amino acids to experimental design to how to read a graph or a Western blot. Lots of important stuff. So definitely refresh if you felt fuzzy anywhere in there. If you like this video, give us a like down below and subscribe to our channel. And comment exactly what type of walkthroughs and videos you're looking forward to next. If you like this, go check out Vikram's 528 Strategies series. Super helpful strategies to help you get your maximal score. And also click the link in our bio to sign up for MCAT practice question of the day so you don't miss out on any test prep before test day. Happy studying and great work.